I am Ikenna Kingsley Uchendu, a lecturer with the Department of Medical Laboratory Sciences, University of Nigeria. The title of my presentation is Methanolic seed extract of Nigella sativa truly protective against toxic chemicals or harmful to tissues? Introduction. Foods or edible plant materials with medicinal properties have been used for treating various diseases for many centuries in folk medicine. Nigella sativa, also known as black seed, is native to Southwest Asia, with inclusions of Pakistan and India, and belongs to the botanical family Ranunculaceae. Traditional uses of this seed ranges from soothing wounds to remedying cough, eczema, diabetes, inflammation of the bronchi and toothaches, and this points to substantial tissue effects. This is the image of Nigella sativa seed. This is the image of cadmium chloride salt that was used as a toxic chemical in the research. The aim of this study was to investigate the protective effects of methanolic seed extract of Nigella sativa against cadmium-induced biochemical and histomorphological alterations in heart, kidney, and liver tissues of albino rats. Methods. Fresh samples of Nigella sativa seeds were obtained from a local market in Enugu, Nigeria. The preparation of the methanolic extract was done as described by Babe et al. With minor modifications. Procedures outlined by Trees and Evans were employed for the phytochemical analysis. This study was approved by the Animal Ethics Committee of the University of Nigeria Teaching Hospital. Experimental design. A total of 25 albino rats were used for the study, and they were grouped into 5 groups: Group A, B, C, D, and E. Group A served as the normal control and received no treatment throughout the experiment. Group B served as a negative control and received cadmium chloride 5 mg per kilogram intraperitoneally for 14 days. Group C served as the experimental low dose and received cadmium chloride and the low dose of the extract 300 mg per kilogram oral for 14 days. Group D served as the experimental high dose and they received cadmium chloride and high dose of the extract 600 mg per kilogram oral for 14 days. And Group E served as a positive control and had cadmium chloride and a standard antioxidant drug, vitamin C, 200 mg per kilogram body weight oral for 14 days. After 14 days, the animals were sacrificed under anesthesia. Blood samples were collected for biochemical analysis, and the heart, kidney, and liver organs were harvested for histopathological analysis. This is the result of the preliminary phytochemical analysis. The results show that alkaloids and flavonoids are present in abundance in the methanolic extract. Tannins and phenols are present in moderate amounts, while glycosides, saponins, terpenoids, and steroids were absent. This is the result of the biochemical analysis. From the results, cardiac markers and the biochemical markers of kidney function and liver function are all shown on the table. From the result, the group that received only cadmium chloride showed significant elevations in these biochemical markers when compared to the normal control group. However, when the negative control group was compared to the low dose extract, high dose extract, and vitamin C separately, the results showed that the low dose of the extract showed a significant decrease in this biochemical marker, showing protection. The high dose extract also showed decrease, but the decrease were not as significant or not as efficacious as the low dose. Now, the low dose extract interestingly showed similar protection as the vitamin C, as seen in the results. This is the photomicrograph of the heart sections among the experimental groups. From the results, it can be seen that the group B, the cadmium chloride group, showed serious damage to the heart's cardiac fibers when compared to the normal control group. However, when group C, the low dose of the extract, and group E, vitamin C, were compared to the cadmium chloride group, it can be seen that there is significant protection in the cardiac fibers, and also the high dose of extract showed protection, but this protection is not as seen in the low dose and in the positive control group. Similar results was also seen in the photomicrograph of the kidney. Low dose of the extract and the positive control vitamin C showed better protection than the high dose of the extract against the cadmium chloride group. The same was also seen in the photomicrograph of the liver, where low dose of the extract group C and positive control vitamin C showed better protection than the high dose of the extract group D as against the negative control group B, cadmium chloride group. Discussion. Accumulation of toxic substances such as drugs, heavy metals, poisons, etc. can cause significant damages to the histomorphology of vital organs such as the heart muscles, kidney, and liver tissues. Exposure to cadmium could produce serious adverse effects in humans via induction of oxidative stress. The results showed that methanolic seed extract of Nigella sativa offered a marked ameliorative effect or protection against cadmium. We, however, observed that the extract, particularly the low dose, acted in similar way as the standard drug vitamin C, which is a known antioxidant. Our observation agrees with the reports of Abukada, who reported that at 500 mg per kilogram body weight, oral administration of Nigella sativa became toxic to the rat used for the study. And in our study, low dose of the extract was 300 mg per kilogram and high dose of the extract was 600 mg per kilogram. Conclusion. The results offer scientific evidence that at low doses as used in foods, Nigella sativa seed has no harmful effects and could very well be potentially tissue protective against harmful chemical toxins like cadmium or drugs. However, higher doses or prolonged use of methanolic extract of Nigella sativa in herbal preparations calls for caution in patients with underlying cardiac, kidney, or liver dysfunction. Thank you.